Sorghum is the 5th biggest grain crop in the world. It is a staple food for millions of people. Sorghum can survive and produce good harvests during prolonged heat and drought. But a deadly fungus that causes sorghum anthracnose can destroy a harvest. Researchers at the University of Johannesburg have found out how friendly bacteria help the plants to defend themselves much faster and more strongly against fungal attacks. This is a low-cost way of protecting food plants and can be used for other diseases and crops in the future.
We investigated the effect of the plant beneficial rhizobacteria, or it's also called PGPR for short, on the innate immune system of sorghum plants. PGPRs, in certain cases, have the ability to trigger and enhance defense response within the plant. The bacteria interacts with the plant root system, set up a chemical communication system, and that leads to the ability of the plant to sense potential pathogens earlier and also to launch a more effective and stronger response against the attacking pathogen. That way the plant expends less energy towards defense and more energy can go to grain production. In nature, in the field, crop plants like sorghum cannot move away from environmental stress conditions. They have to cope with what they have. So plants have an innate immune system, and this innate immune system allows them to survive and proliferate in the immediate environment. This innate immune system is quite often based on chemicals within the plant, and we can refer to them as a chemical cocktail that can be changed according to conditions. So when a sorghum plant is attacked by a pathogen like the Coleotrichum subplunihalum, it turns on its innate immune system. But quite often that might not be sufficient, and the plant has to expend more energy to survive, and quite often the grain yield is then less, or the plant might even die in the process. So in our research, we looked at ways and means to enhance the immune system of the plants so that a susceptible plant can become tolerant, a tolerant plant can become resistant, eventually yielding more grain that can contribute to sustainable food production. In this specific example, we used what is known as plant beneficial bacteria to form an interaction with the plant roots, leading to a chemical communication system between the bacteria at the roots and the plant shoots that is encountering the pathogen attack. That chemical communication was then investigated by means of metabolomic tools and approaches using chemometric analysis.
In this study, we generate big data, gigabyte size, that are so complex. So we employ chemometrics, machine learning methods to extract information from such big data sets. So the information we extracted shows us that the primed plants change the chemical defense for a stronger and effective security system. Changes that were observed included increase in the levels of lipids, increase in the level of amino acids such as tryptophan, and also the changes in flavonoids, which are chemicals that the plant uses to defend itself.
So we made a significant amount of progress towards understanding the phenomenon of bacteria-induced priming and associated crop production. Our findings can also be extrapolated towards other crops and other pathogens under different environmental conditions.